Space birds, tardigrades as the tiny titans of astrobiology and beyond. Hey everyone, my name is Kia and today we're gonna talk about tardigrades and have lots of fun. First, we'll introduce tardigrades, then we'll talk about cryptobiosis and anhydrobiosis and the molecular mechanism involved, then we'll delve into significance of tardigrade in astrobiology, and finally we will go over their unique ability to protect DNA against radiation. In the end, we will summarize the presentation with an overview of everything we've learned. Introduction. Tardigrades typically have 8 legs, they feature a circular mouth, and have an omnivorous diet. In some graphics in the presentation, you'll see that instead of the mouth, they have a nose-shaped mouth, but this is only to show that they look like other animals like birds. Not accurate. I should also mention that the phylum Tardigrada belongs to the Panarthropoda, meaning they are closely related to Arthropoda and Onychophora. Also, here's a real-life image of a tardigrade, except for the Hawaiian shirt. Tardigrades look like birds, that's what they're called, water birds, and they're among the most resilient animals on Earth. They can survive in extreme cold down to negative 273 degrees Celsius, extremely hot up to 150 degrees Celsius, radiation levels that are deadly to most animals, and even space vacuum. Tardigrades inhabit many environments, from deep oceans to Antarctica extremes, and even in urban mosses. Measuring just 50 to 1,200 micrometers, they exhibit eutelie, meaning that they are born with a fixed number of cells, and the cells just grow in size as the animal grows. Cryptobiosis. Tardigrades master the art of cryptobiosis, which is a reversible suspension of metabolism in response to environmental extremes. This state manifests as various forms, like anhydrobiosis, which is enduring desiccation, cryptobiosis, which is surviving freezing, and anoxybiosis, which is withstanding anoxia. Unlike many other organisms that are closely related, like crustaceans and many insects, tardigrades can enter these states at any stage in their life. The molecular mechanism of anhydrobiosis, which is withstanding desiccation, is divided into 4 subsections. The first subtype involves water replacement, in which the sugar trehalose replaces water during desiccation. This prevents cell damage and keeps structural integrity. Second subtype of anhydrobiosis centers on damage repair and molecular shielding. To resist desiccation, tardigrades use various bioprotectants, like heat shock proteins or HSP and late embryonic abundant or LEA proteins. Tardigrades begin transcribing heat shock protein genes as they are going into the anhydrobiosis stage, and the mRNA produced is subsequently translated into active heat shock protein after the rehydration. So upon rehydration, HSP and LEA proteins come into play, assisting the reconfiguration of proteins into their functional forms. Moreover, intrinsically disordered proteins, or DIP, act as a molecular shield that safeguards essential molecules in stressful conditions. The third defense category is protection and DNA repair. Dehydration leads to rise in ionic concentration and reactive oxygen species that can damage biomolecules like DNA. To combat this, tardigrades fine-tune their antioxidant processes and also use special proteins, notably the damage suppressor protein, or DSUP. We're going to talk much more about DSUP in this presentation. The fourth and final subtype involves mitochondrial protection. LEA and DIP, which we discussed before, also play a role in keeping mitochondrial integrity during desiccation. This ensures that tardigrade survival in extreme conditions, as mitochondrial function is needed to produce energy so the animal can survive. Astrobiology significance. In September 2007, tardigrades went on a 12-day journey with Photon M3 spacecraft. This mission exposed them to unique space conditions, including microgravity and cosmic radiation. This provided an opportunity to study the effects of desiccated tardigrades in the space conditions. The study by Rebici et al. 2009, the continuation of the same study, used 2 types of desiccated tardigrades. One group naturally desiccated in leaf litter, F1, and the other group desiccated on paper in laboratory condition, represented as F2. And the controlled group, TC4, were kept on Earth, mirroring the temperature profile experienced in space mission. The key findings show that both groups of desiccated tardigrades exhibited impressive survival rates in space. We can see F1 has a survival rate of 94.4%, F2 had 78.9%, with TC4 58.9% survival rate. Remarkably, tardigrades in space molted, laid eggs, and hatched, with the newborns displaying normal morphology and behavior. Also, some tardigrades post-space mission showed a lifespan of 5 months on Earth while actively reproducing. Based on their documented tolerance, tardigrades were the first animal to survive at the exposure to space conditions. The resilience physiology in outer space offer insight into what extraterrestrial life might be like. Also, understanding and applying their physiology can revolutionize human space exploration by improving astronauts' resilience. Further, I want to emphasize that replicating experiments like the one that we've seen on international space stations is essential for a deeper understanding of life's adaptability in space. There's a significant gap in applying the insight gained from tardigrade physiology to human biology, so research could explore how tardigrades-like features can be mimicked in human biological system that could enhance the space missions, as humans can be more resilient to space conditions. To add to that, while some unique proteins like LEA and IDP are identified, and the structure and specific function of many of these proteins still are unclear. So in-depth protein chemistry and structural biology studies are essential to show how these and other unidentified proteins contribute to tardigrades. DNA protection. But what is the secret to this incredible resilience? At the heart of this mystery lies a remarkable protein called DSUP, damage suppressor protein, which was mentioned previously. We know that intense radiation can damage DNA and lead to cell death, cancer, and malfunction. The discovery of DSUP in Rhamazarius varionatus, a species of tardigrade, in 2016 opened up exciting possibilities. Ricci et al. 2021, it's a relatively new study, investigated the role of DSUP in protecting human cells against oxidative stress and ultraviolet radiation. The experiment began with the transfection of HEK293 human cell lines as the researcher introduced the DSUP gene into the cells. They exposed both DSUP plus and non-transfected cells to hydrogen peroxide for oxidative stress and UV radiation. The team then measured cell survival, DNA damage, transcription factor activation, and gene expression changes to evaluate DSUP's protective effects. So the result of the study showed that DSUP plus cells had remarkably higher survival rates compared to non-transfected cells when exposed to both hydrogen peroxide and UV. So the study found that in the presence of hydrogen peroxide, DSUP physically protects DNA in the cell. It also activates several detoxification pathways, which are aiming to remove intracellular radicals. Unlike the action against hydrogen peroxide, in UV exposure, DSUP does not provide direct physical protection to DNA. Instead, it appears to boost the efficiency of DNA damage repair mechanism. This enhances repair capability, repairing cyclobutane pyrimidine dimers, which are a common type of DNA damage caused by UV radiation. These findings have exciting implications as they could be pivotal in protecting astronauts from cosmic radiation, and they can also pave the way for innovative cancer therapies.
Yeah! Who the hell are you?
Sorry, didn't mean to scare you, little buddies. My name's Vernon. I'm a water bear. Well, here she is. Home sweet home. Everybody, we've got guests.
Water.
That's right, and we need to get back there. And if you guys help us, we'll make sure you never want for water again. What do you...
It's the dust mites.
Here's the microboard. I just need to reroute the circuits. Hold. Hold.
Now! You never told me what to do.
Jump! Why? Trust me! Let's take a look.
So in today's presentation, we journey through the extraordinary world of tardigrades, from their fascinating biology to their implication in astrobiology. We started by delving into the resilience of these macroanimals. We focused on the survival strategy in extreme environments via cryptobiosis, which also encompasses angiobiosis, and examined the molecular mechanism underlying these capabilities. We then mentioned the significant milestone of tardigrades surviving in space, which offers insights into potential extraterrestrial life forms. Our exploration, however, revealed a key knowledge gap. It is yet to be studied how to translate tardigrade resilience mechanisms to human biology for space exploration. Moving forward, we highlighted the need for ongoing research on the International Space Stations and deeper analysis of tardigrade proteins. This can have potential implications for human space travel and medical science. We also discussed the significant role of DSUP in protecting DNA from radiation and oxidative stress, which underscores the potential application in space and healthcare. In closing, we recognize tardigrades not just as subjects of curiosity, but as beacons guiding us toward new scientific horizons. Their study is key to unlocking the secrets of endurance and adaptability in cosmos. Thank you for attention, and thank you for joining me.